So I've had the pleasure of working with Ayaan on RNA therapeutics. So our topic was understanding kind of how we can use RNA in order to improve human disease conditions. And we started off by really kind of doing a deep dive on RNA, kind of its chemistry, what even sets it off from other therapeutic compounds. And then we were for a while discussing and kind of brainstorming about what are the different contexts where this type of therapeutic molecule could be useful. We talked a bit about cancer, and we also talked about the kind of work that we want to do. So we explored some data sets and talked about whether we want to do analysis and or literature review. And ultimately what we decided to do was to do a review of the potential of RNA therapeutics for the neurodegenerative disease landscape. So that's what you will be hearing about today. Basically, Ayaan reviewed, on the one hand, what is kind of our RNA-based toolbox that we have, and then what are actual neurodegenerative diseases, what are their mechanisms. And then he kind of like brought this together in order to propose actually some new RNA therapeutics that could, you know, maybe in the future be developed. And yeah, he's been doing a great job over the past weeks, and I look forward to his presentation.
So my presentation is going to be on exploring the potential of RNA therapeutics for neurodegenerative diseases. So RNA therapeutics have largely broadened over the last decade or so, as in researchers have found many new RNA molecules and methods to utilize them for therapeutics. And this report aims to outline some major neurodegenerative diseases that have been plaguing society, and then using these therapeutics to combat them. So I'm going to be dividing my presentation into 4 parts. The first is going to be an introduction on all of this. Then I'm going to be talking about RNA therapeutics and their mechanisms in detail, then the neurodegenerative disease landscape, and finally, I'm going to be talking about utilizing this toolbox to combat neurodegenerative diseases as a whole. So starting with the first on my introduction. So the central dogma of biology relies on the interactions between DNA, RNA, and the protein. So DNA, like we all know, is responsible for providing the basic coding for a protein. And what many people don't know is that there's actually a middleman over here called mRNA. So DNA is transcribed into mRNA, which is then finally translated by something called a ribosome in the cell into a protein, which can perform crucial biological functions. And this is the central dogma for biology. Now I'm going to talk about therapeutics in general. Based on this, there are only 2 prevailing therapeutics that we're using today, DNA and protein-based therapeutics. So the thing with DNA-based therapeutics like CRISPR-Cas9, they're very permanent. So that may be good for some diseases and not beneficial for others. And protein-based therapeutics are very good for modifying the functions for other completed proteins, but the development is super costly. Like it could take like a decade to come up with a fully functional protein therapeutic and a lot of money. RNA, the middleman in the central dogma of biology, is the newest innovation in the field of therapeutics, and I'm going to be talking about why it may be the most useful, combating neurodegenerative diseases. Now onto a little bit about why they're good. So being intermediate, it has the benefits of DNA and proteins, but it kind of removes the harms of both of them. So it's programmable. So the thing with the RNA and DNA, they're both based on only 4 chemical bases. So it's really easy to just pick and choose what exactly the structure of them has to be. And through this, it doesn't really take as long to create RNA therapeutics compared to protein therapeutics. So it's like the COVID-19 vaccine, which was an mRNA vaccine. It only took a year for to be created, while other vaccines could take a lot more time. mRNA and proteins are both transient molecules. So that means it's not as permanent as, say, a DNA therapeutic, which would permanently change your genome. It's rather a more temporary effect and based on multiple medications. So now on to the first part is on targeting the problem, which is neurodegenerative diseases. So 1 in 6 people are actually affected by a neurodegenerative disease, according to UN statistics. So it's a really big problem today. So the insidious nature of neurodegenerative diseases, 4 pieces of characterization on what they actually look like. The first thing between all neurodegenerative diseases is that they all have some sense of memory loss, difficulty in movement, so motor loss, changes in mood, behavior, and speech and language problems, and there's also numbness. they're all they're all progressive in nature, as highlighted by degenerative. So they all have the capability to just keep getting worse until there's like fatality or like some real serious level of brain impairment. They're all incurable. So these diseases usually have only hospice care and pain relief as medications. So like protein and DNA therapies aren't really ideal for them, because DNA and protein based therapeutics, like neurodegenerative diseases, are really hard to predict, and because they're late onset and these diseases are progressive, and you need dynamic treatments for that. DNA therapeutics are like mostly based on genetics, and since there's environmental factors here, it's really difficult to like properly design a therapeutic purely based on one specific gene, because it depends on the patient. And there's also like, since these diseases are progressive, like it's dynamic treatments, DNA is not really a dynamic treatment. One may assume that protein therapies would be better than, but they're really difficult to design for such a complex machinery, and like so far they've really failed in this particular area. So now I'm going to be talking a bit about the RNA-based tools that we have, our RNA toolbox. Four RNA tools that I'm going to be talking about today. The first is going to be like mRNA, the most popular, messenger ribonucleic acid. The next one's going to be siRNA, small interfering RNA. The next one's microRNA, and finally we have antisense oligonucleotides. So starting off with like messenger RNA. So messenger RNA is responsible for the creation of a protein. So what happens is a protein looks at the DNA strand and makes a copy of it, a single-stranded copy of it, and that copy is called the mRNA. The mRNA is then taken to the ribosome, which is like the small piece of machinery in the cell, and that turns it into a protein. The Moderna COVID-19 vaccine manipulated the mRNA so that it could induce an immune response. So it introduced mRNA that would make viral antigens, and it went into the cell. The cell generated harmless COVID-19 parts, and the immune system got activated, and that was how they managed to create the vaccine, which in only 1 year. So the manipulation of mRNA can allow us to make complex proteins quite easily. Next, we're going to be talking about siRNA. So on the left, you'll see like a diagram of how exactly siRNA works like naturally. It's a little bit complicated, so I won't go into that too right now. But basically, siRNA is responsible for complicating the central dogma. So of course, there's DNA, mRNA, and protein. So siRNA comes in the part where there's a difference between mRNA and protein. So what it does is it cuts the mRNA, right? So siRNA, once it's bonded with this thing called a RISC complex, it goes and it cuts the target mRNA, which it bonds to. And after it destroys it, they're all degraded by something called a ribonuclease, which is a special enzyme. And when it does this, basically the ribosome can no longer read the mRNA, and you prevent the creation of the protein. So the good thing about siRNA, it's very sequence specific, so it won't end up destroying some other protein, which would be really bad. It's just very easy to program and design new therapeutics with siRNA. Next, you have miRNA. Here, this is like on the left, you have a diagram which is very similar at the end, RISC miRNA complex. This is a laboratory setting, so this is how they would make it in the lab. But I'm not going to go too much into that. But very similar to siRNA, miRNA can interrupt the central dogma for biology. It basically just cleaves the target mRNA. But however, miRNA is capable of cleaving many mRNA strands, which is what mainly distinguishes it from siRNA. So like it doesn't need a full complementary strand. It can literally like it can stop the production of multiple different types of proteins. So this is really good for like a multifaceted protein disease. Next, you have antisense oligonucleotides. So they have two ways of reducing the protein of interest. The first thing they can do is bind to the complementary mRNA and attract ribonucleases that will destroy it. And the second method of mRNA blocking is steric hindrance. So the AGO, antisense oligonucleotide, bonds to the mRNA, and as the ribosome, one could say, is scanning it, it skips that particular part because it's already bonded, and basically it edits the final protein outcome without that particular part of the mRNA content. Now I'm going to move on to chapter 2, neurodegenerative diseases. So there's a couple commonalities in neurodegenerative diseases. The ones we've chosen basically rely on the concept of defective proteins. So proteins, when they're misfolded or defective inherently, they can clump together and they can cause really large aggregates, and that causes neuronal death. And then as more neurons die, it becomes degenerative and you lose more memories and you get loss of motor function. So that's how these diseases are degenerative. So most of these diseases are hard to predict based on genetics purely, because there are environmental factors, and neurodegenerative diseases tend to occur late onset. So even if you could like do something with DNA editing where you would do it in the embryo, these would not be good enough targets for gene editing. um, Gene editing is currently done mostly on patient cells extracted, engineered, and returned into the patient. But like it's really hard to do that with neurons because they're in the brain and it's a really sensitive area. And is cell therapies for neurons is extremely complicated, expensive, and time consuming. Additionally, DNA modifying therapies aren't dynamic. While these diseases are very dynamic, they keep changing. So it's really important to be able to keep up with it, or else the treatment will fail. And protein therapies are very expensive and difficult to design and have only been able to provide hospice capabilities till now. We researched on a lot of diseases, so like the complete list includes these on the side, but we chose 3 diseases in particular: Parkinson's, Alzheimer's, and spinal muscular atrophy, for a couple of reasons. So in Parkinson's, it's caused by like multiple mutations, so like all of these genes. Buildups of something called alpha-synuclein protein causes the neuronal death. So this alpha-synuclein protein, it's mutated and then it creates aggregates which kills many neurons. And the interesting thing about this protein is that it can cause like non-mutated versions of these proteins to misfold or like become mutated. So this is like infectious within the patient's brain, but it can't spread to another person around you. Next is Alzheimer's, which is caused by a buildup of 2 proteins, so mutated amyloid beta and then hyperphosphorylated tau. So both of these become insoluble, and amyloid beta and tau proteins make plaques and neurofibrillary tangles, respectively, which will all lead to cell death. Finally, spinal muscular atrophy, which is really, really interesting. So in humans, we have 2 genes, in this case, which is SMN1 and SMN2, which is we're the only organism who have 2 genes like this. So basically, when there's a SMN1 gene mutation, there's like a defective SMN proteins, and that leads to motor skill defects. The SMN2 gene also generates SMN proteins. However, it's been found that those proteins are kind of defective in the sense where they degenerate far too fast. However, like on the off chance, sometimes the SMN2 does generate good good proteins, SMN proteins. And in a spinal muscular atrophy patients who had higher expression of the SMN2 gene, it was found that their symptoms were less profound. However, the SMN2 gene was far too defective to actually be able to combat the SMN1 gene mutation. So going into detail on Parkinson's disease first. So Parkinson's disease has been, it's a very common disease, and it's been causing motor dysfunction in millions of people. And since it has like many genetic factors, like we saw on this slide, we decided that microRNAs are an ideal therapy. So microRNAs, of course, are capable of targeting many genes, which is why we chose them for Parkinson's. So already microRNAs are present in the human body and have important regulatory functions. And there's something called microRNA 7, which is found in brain, which is naturally found, but it's in the body. But it's been found in patients with Parkinson's disease to have less levels of microRNA 7, suggesting that this microRNA is causally implicated in alpha-synuclein accumulation. This means that the more microRNA 7 you have, the less likely you are to have this accumulation. So in Parkinson's disease patients, they have very little amounts of these microRNA 7. So we could artificially synthesize this microRNA to combat the protein aggregation and inject it into the patient as a method of therapy. Next, we have Alzheimer's disease. So there's 2 proteins that cause Alzheimer's disease. So you need like a combined strategy. So the first idea is to reduce amyloid beta. So Alzheimer's is caused by a buildup of amyloid beta, which is mutated. And so there's an enzyme called neprilysin, which can actually degrade amyloid beta. So all enzymes have cause proteins. So delivering neprilysin mRNA will degrade amyloid beta. How? So first we'll design mRNA to make neprilysin in the lab. And then once you insert this into the cells will begin generating this enzyme, and that will in turn degrade the amyloid beta. Alternatively, siRNA could be used to reduce beta secretase or gamma secretase. So if you see in this diagram in the top right, beta secretase and gamma secretase are responsible for the creation of amyloid beta and splicing it. So if you remove either one of them, what will end up happening is you will not be able to get amyloid beta. So reducing them using siRNA would lower amyloid beta levels, which would in turn benefit us in terms of therapy. The second strategy is to reduce tau proteins to remove the neurofibrillary tangles. So delivering siRNA, which is complementary to the mutated tau mRNA, would be the method that we would use to combat tau neurofibrillary tangles. And a combined version of both of these therapies would be very beneficial in my opinion. The only limitation to this therapy is that it would cause a reduction in non-mutated proteins. What this means is that since there's like mutated amyloid beta, but there's also normal amyloid beta, which is important, same for tau. But these methods would lower their levels as well. So, I mean, it's kind of like a weighing situation where it's maybe more important to stop Alzheimer's than have lower levels of these proteins. Moving on to finally the last disease, my favorite, spinal muscular atrophy. So there's three things that could happen here. The first is mRNA replacement therapy, so that you can have correct SMN protein formation. So this looks like inserting mRNA that would generate SMN protein, so that you could combat the effects of the mutations in the SMN1 gene. The second option is to modulate the splicing of SMN2 RNA with antisense oligonucleotides. So when the SMN2 gene is being transcripted into a RNA molecule, the antisense oligonucleotides can come in and remove the defect. So like we know that there's an inherent defect in the SMN2 which prevents it from generating good SMN proteins. So you could use AGOs to come in and change that and make them work so that they can be effectively a replacement for the SMN1 protein. The third option is to degrade the incorrect copy of the SMN, example like with siRNA, or sterically block the start site of the SMN1 mRNA. So what this basically means is that the first option is to degrade the mRNA that comes from the SMN1 gene, or the second one is to take antisense oligonucleotides and make them block the start site where the ribosome would usually attach, and effectively stopping the protein generation coming from SMN1. So your options for combination therapy would probably be 1 and 3 or 2 and 3. Either way, you'd be getting more of the correct protein and less of the incorrect one. With that, I'm completed my presentation. Thank you, Mr. G, for organizing this, and thank you, Arianna ma'am, for walking me through, because this was there were a lot of complications while I was making this, so it's very useful.